Okay, so here we're going to start talking more microscopically about the nephron. Okay, so if you look at this and you look at the kidney, okay, here's the cortex. Oops, shoot, I missed. And what we're going to try to do is we're going to magnify that little piece and talk about the parts of the nephron. Okay, so remember, the nephron's actually where the urine is going to be formed. Now, we don't call it urine yet, okay, so we call it filtrate at the beginning. We don't call it urine until we get to the collecting duct. Okay, so we don't call it urine yet. We're still calling it filtrate. Okay, now, there's two parts. The way we look at the kidneys, we look at it two ways. We look at it as the filtrate and how the filtrate is formed, but we also look at it from a blood perspective. Okay, so we're looking at it from the vessels, and we're looking at it from how we make urine. Okay, so the nephron is this series of tubes. Okay, so what it's made out of, okay, first thing is this thing called the renal corpuscle. Okay, so the corpuscle is kind of like a bump or a circle. Okay, and it's this part right here. So this is the renal corpuscle. Okay, and the renal corpuscle is made out of this thing called the glomerulus. Okay, and I think of the glomerulus kind of like a fist. Okay, so if you can picture a fist, okay, so think of it kind of like this. And then it's actually going to be covered. Okay, so here's what's happening. Okay, so we have blood coming in, and then it's going to get filtered through this thing, through the glomerulus. Okay, so the glomerulus is a kind of like a, if you look at it, it's the red thing here. So it's this tangled up mess of capillaries. Okay, and that's where we're going to filter the blood through. Okay, so there's thousands of these glomeruli all around the edge of the kidney. Okay, so they're all around here. I hope that makes sense. So they're all around there. And what's going to happen is the blood is going to come into them, and it's going to be under a constant pressure. And then we're going to filter the blood, okay? And we filter the blood through this glomerular capsule. So picture like putting your fist into a Ziploc bag, okay? And then the urine is going to come out this way. Okay, so the blood comes in, it gets filtered through here, through this area. Okay, and then the urine's going to come out. The rest of the blood is going to continue going this way. I hope that makes sense. Okay, now, the path that the filtrate is going to follow is it's going to go through some tubes. Okay, so these renal tubules. Okay, so it's going to go through, the first thing is the proximal convoluted tubule, and we always abbreviate that PCT. So proximal means it's close to the glomerulus. Okay, so it's proximal to this bump, to this circle. Okay, distal means it's further away from the glomerulus. Okay, so the distal convoluted tubule is DCT. Okay, so proximal convoluted, okay, so if something's convoluted, it's just a mess, okay, so it's like think of a tangled up ball of string. That would be convoluted. That's the way these are. Okay, so they're just these masses of tangled up tube. Okay, now if we trace the flow, okay, which we have on the next slide, what we're going to do is when we make urine, okay, we're going to go through the glomerulus, through the PCT, the proximal convoluted tubule. Okay, then we're going to go down this nephron loop, and it's got a descending limb and an ascending limb. Okay, then we're going to go through the distal convoluted tubule. Okay, and then we're going to end up in this collecting duct. And more than one nephron can dump into a collecting duct. Okay, now the way that the blood flows, let me use red for that, is the blood, and we'll talk about this, it's going to come in the afferent arteriole. Okay, so it comes in the afferent. It goes into the glomerulus. Okay, and then it exits the efferent. Okay, so A before E. So it enters with the afferent and it exits with the efferent. The afferent is bigger than the efferent in diameter. Okay, so the afferent has an increased diameter. It's bigger. Okay, and the efferent is small. So if you think about that, that's the idea behind that, is to keep the blood pressure constant in there and keep it under pressure. Okay, now the layers of the glomerulus, well, let's talk about them on another slide because this is getting kind of busy. Okay, so here it is. Okay, so here it is all stretched out, laid out. It makes it easier to see, although it's not really anatomically correct. So anatomically, it's that tangled up, convoluted mess. But this is way, way, way easier to see. So this is a picture from your book. So here's the glomerulus. Okay, they call the two ends of the glomerulus. Okay, so the end with the blood vessels, they call the vascular pole. The end with the tube, they're going to call the tubular pole. Okay, so the afferent arteriole enters in. Okay, the efferent arteriole exits. Okay, there's the glomerulus. So that's kind of like your fist. Okay, and it's got layers of cells on it, which we'll talk about. Okay, now the glomerulus is covered with this capsule. Okay, so the capsule, let's make it blue. The capsule covers the glomerulus, and there's space where the urine's going to get collected. Okay, so I'll have to show you. I always like do a thing with my hand in a bag. Okay, so if you picture putting your hand inside of like a Walmart bag or a Martin's, one of those plastic bags. Okay, so you're not putting it in the opening of the bag, but you're putting it in within the bag. So the part of the bag is touching your hand. Okay, and then there's kind of be space, and then there'll be an opening. I'll try to find another picture for you. So it kind of goes like this. Okay, so if there's your glomerulus, this covers it like this. And this is space where the urine's going to collect. And you can see that on the models in lab. Okay, now this whole thing of the glomerulus and the capsule, that's called the renal corpuscle. And that always sounds like something that you should pop, doesn't it? Corpuscle, it should be something like you're picking at. Okay, then we have the proximal convoluted tubule, okay, the nephron loop, and then the distal convoluted tubule, and then the collecting duct. Okay, the nephron only goes to here. Okay, this is where the nephron stops. The collecting duct is not considered part of the nephron. Okay, more than one nephron can dump into here. Okay, so we might have other nephrons dumping in, like their DCTs could be dumping in to a collecting duct. Okay, now on here it's important to see the cells. Okay, so the histology, what's going on. Okay, so what I want you to notice, and I know I have this written down in some other slides. Okay, so this is simple cuboidal epithelium, but it has what we call a brush border. Okay, now the brush border, it's a whole bunch of microvilli on the cells, And anytime we have that, that's going to increase the surface area. So if we increase the surface area, that's going to allow for more transport. So increase surface area, increase cell transport. Which that makes sense, right? So in here, this little blue, that's increasing the surface area. Those are the microvilli. Okay, so PCT, proximal convoluted tubule. The nephron loop is unique because of the way that it's formed. So it has two different segments. It has a thick segment and it has, so here's thick. Okay, so it's thick from here to here and down here. Okay, and then it's thin down here. Okay, now the thick segment is going to be not permeable to water. I have this in the other slide. Okay, so thin segment, let's put water permeable. So water can go through this, through the thin, but it can't go through the thick. Oh, shoot. No. Okay. No water. So we'll talk about that more. Okay, so, and the naming of the nephron loop is really easy. So it's the descending limb is going down, and the ascending limb is going up. So that's like the easiest thing. They used to call the nephron loop the loop of Henle. Okay, so it used to be named after a guy, but now it's not named after him anymore. Okay, and then the distal convoluted tubule is the end, and you can see the microvilli on there. It doesn't quite have as many. Okay, now we will talk about the parts, the intercalated discs, the principal cells. We're going to talk about those parts of the collecting duct as we go on. Okay, so I hope that makes sense. Okay, now here's the text that goes with that last slide. Okay, so the PCT is the longest part of the nephron. Okay, so it's the most coiled. Okay, so it has the most transport going on. It's the most metabolically active. It's going to do most of the work. Okay, so I just put what I just told you. So simple cuboidal cells, it's got brush border, that increases the surface area for absorption. Okay. The descending and ascending limbs of the nephron loop. Okay, so it has a thick segment, which is simple cuboidal, okay, and a thin segment, which is simple squamous. Okay, the thick segment is in the very first part of the descending limb. Okay, so if I draw it, okay, so thick up here, okay, and thick here. Okay, so all of this is thick. Okay, it is permeable to ions. Okay, so I have active transports of salts, so ions. Okay, the thin is all of this. Okay, and it's permeable to water, but not salt. Okay, so they do the opposite things, and it'll make sense eventually as we go on. Okay, so I hope this makes sense. So thick, thin. Okay, and then the very last part is the DCT, so the distal convoluted tubule, which is cuboidal epithelium, but it doesn't have many microvilli in it. So most of the transport is occurring before we get to the DCT. But the DCT, it has some important features. That's actually where the hormones work. It has hormone receptors. Okay, so this is the end of the nephron. Okay, so all of this is part of the nephron. Okay, so now there's two flows that we're going to talk about. So you need to know how urine's made, and then we have to know the blood flow. And you're going to work on these in lab too. Okay, so the flow of the glomerular filtrate. Okay, so we're going to start at the capsule, the glomerular capsule. Okay, then we're going to go through the proximal convoluted tubule. Then we'll go through the nephron loop, the two parts. Okay, the descending is first, and then the ascending. Okay, then we go to the DCT. Then we'll go to the collecting duct. Okay, so there's the collecting duct. Okay, then we'll drop into the papillary part of the pyramids. Okay, then we'll go to a minor calyx, and then we'll jump into a major calyx. And I lost my arrows, and I thought I saved this different. Okay, then we go to the renal pelvis, out the ureter to the bladder. So there should be like commas in there. To the bladder, and then out the urethra, and that's a bye-bye PP. So I hope you're gonna know this. You're gonna be one with that. Okay, now the blood vessels around the nephron. Okay, so if you look at the nephron, okay, there's two kinds of nephrons. Some are short and some are really long. Okay, so the short nephrons are called cortical nephrons. Okay, so most of the time they're in the cortex. Okay, so they're in the cortex out here. Okay, so they're out here and they only go down into the medulla a little tiny bit. Okay, so 85% of the nephrons are cortical nephrons. Okay, now the other kind of nephron are called juxtamedullary. Okay, so juxta, so they're close, juxtaposition, they're close to the medulla. Okay, so these guys are going down into the pyramids. Okay, so if you look up here, they're going to be going down into like this part. Those are the juxta. I hope that makes sense. That's the juxta medullary. Okay, only 15% of them go down into the medulla. Okay, now they have very long nephron loops. Okay, so they're going to go way down in. Okay, and what they're going to do is they're actually going to maintain the salt gradient in the kidney. So the salt gradient in the kidney is going to be important for water conservation. So this is where all the osmolarity and hypotonic come in. Okay, so these guys, they're going to conserve water for us and maintain a salty concentration in the medulla of our kidneys. Okay, now, another thing I want you to see, so the blood vessels that are around there. Okay, so blood vessels are, here's the artery that's going to be taking blood to the kidneys and to the glomerulus. And then these are the veins that'll be bringing it back. There are blood vessels that are going to be wrapped around these. Some of them, the ones that are up here, I thought they would be labeled on this picture, but they're not. These are peritubular capillaries. Okay, so they're around the tubes. Okay, so peritubular capillaries, that makes sense, right? And then down here, these ones that are straight, that are wrapped around, these are actually called vasa, so that's like vessel, and recta. So rectus is straight. So vasa recta, these are the straight ones that are flowing down here. Okay, now, the juxtaglomerular apparatus. Okay, now, when we have to have a way to regulate blood flow into the glomerulus. Okay, so this is what the glomerulus looks like up close. Okay, so this is what I was trying to describe to you with the bag. Okay, so under here, this is the glomerulus. So these are capillaries. Okay, and they're covered with a visceral layer. Okay, so here's the visceral layer laying right on top of the capillaries. The visceral layer of the capsule. Okay, then out here, this is the parietal layer. Okay, so visceral's laying right on top and parietal is outside. Okay, there's going to be a space in there. And the space in there is called the capsular space, which that's pretty profound, huh? In that space, that's where the urine is going to collect. Okay, so where those green arrows are, that's where the urine's going to be. Okay, and then the urine's going to flow out the PCT, which we're not calling it urine yet. It's still filtrate. Okay, so parietal and visceral, part of the capsule. Okay, now, the whole thing's the corpuscle, which we went over that. Okay, the artery that's going in, the arterial going in, is the afferent. Okay, and the arterial exiting is the efferent. Okay, now, the way that this is set up, remember, it's not really all laid out all nice and neat like they have it in the pictures with the PCT and the DCT. Really, it's all globbed up kind of like this. Okay, so realistically, this is actually the DCT. Okay, and the DCT is close to these arteries. So they're on the vascular pole of the glomerulus. Okay, so the DCT is close to these arterioles, which seems weird because on the picture, right, we always draw it out like this. Well, we're just drawing it like this so you could see it. In the real world, it's more like this. It's not pretty. Okay, now, this whole part, okay, I'm going to use black. This whole region here, we call the juxtaglomerular apparatus, okay, or JGA. Okay, now, what it's going to do is it's going to regulate blood flow into here. Okay, so these are really, it's a really fine filter, essentially. It's a filter. You could think of it as a water filter. Okay, so it's a really fine filter. And if you force too much blood in through here, you could blow this out and blow holes into it. Okay, and then everything's going to go through and the filter's not going to work. So we want to keep a constant blood pressure going into here. Okay, now, there's two kinds of cells that are going to regulate blood flow through here. Okay, and they're part of the juxtaglomerular apparatus. So the first cells are called granular cells. Okay, we used to call them juxtaglomerular cells. Okay, and the granular cells are found in the afferent arteriole. Okay, so they're in here. Okay, now what they do is they're actually smooth muscle. So what happens is if they get stretched, okay, they're going to contract. Okay, so if the blood pressure goes too high, they're going to contract and they're going to reduce the flow into there. Okay, now another thing that they do is they produce, store, and release our friend renin. Okay, so they release renin. Okay, what does renin do? We talked about renin before, right? And renin activates angiotensin. Okay, now the other kind of cell that we have in here, they're called macula densa cells. And macula densa cells they're epithelial cells, and they are part of the DCT. Okay, so they're up here. And what they do is they're going to monitor the concentration of the filtrate, okay, as it comes through. So it's going to see how much salt is in there. So by monitoring this, okay, they can determine whether blood flow needs to increase or not. Okay, so I always think these are like quality control. Oops, I can't spell. Quality control. So essentially, what these cells are doing is they're monitoring. Okay, so the urine, let's go back here. Okay, so urine's coming through, going through here. It gets to the end. If it gets to the end and quality control says, ooh, you're not the right concentration, okay, it's going to send the message back over here to the arterioles that are entering it and say, you need to slow down flow or you need to increase flow. So essentially, they're monitoring the quality of the urine or the filtrate, and they're going to communicate with the cells in the afferent arteriole to determine what should happen. Okay, now this, as you can see, is kind of complicated, okay, and it's changed over time. So this is the latest model of what they believe is happening. Okay, so it has changed since I've been teaching it. Okay, now, path of blood. So you're going to learn this in lab. Okay, and I have it on the next picture, so I think what I'm going to do is I'm going to go to the picture because it'll be easier to see, but I wrote it out for people that are more linear. I don't know what this little R is. I think this was a dot that got messed up when I put it into this program. So here we go. So blood's going to enter renal artery. Okay, the renal artery divides. Okay, when it divides into these segmental arteries. Okay, so they segmental arteries. Okay, and then they subdivide into interlobar. Okay, and interlobar go between the lobes. Okay, so in between the pyramids. Okay, the arcuate arteries arc around. Okay, so they kind of arc over the pyramids. Okay, and then they feed these interlobular arteries. Okay, so remember, the smaller the part is, the longer the word. Okay, so interlobular arteries. Okay, now the interlobular arteries go to the afferent arteriole. Okay, so they're going to take the blood in here. Okay, and that goes to the glomerulus. And then the blood leaves the efferent arteriole. Okay, then we could go two ways. Okay, we could go around these paratubular capillaries, which are wrapped around the tubes. Okay, or we could go down here to the vasa recta. Okay, so we're really going both ways. Okay, then gas exchange is occurring. Okay, so then what's going to happen is we're going to go back the same way we came. So we're going to go back interlobular veins, okay, to the arcuate vein, to the interlobar vein, and to the renal vein. And that's going to go back to the inferior vena cava and take the blood back to your heart. But this picture, I think, helps because it's a good picture.